Thank you everyone for joining us, and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, June 23rd, 2015. The title for today's webinar is Aiming the Immune System at Brain Cancer. In this webinar for patients and caregivers, we will discuss current immunotherapy treatment options for patients with brain cancer and how close we are to making these treatments available to more patients. Today's webinar is made possible through the generous support of Antigens. Antigens is an immuno-oncology company that believes the best path for treating disease is through activating our body's natural mechanisms. For 20 years, Antigens has focused on building technologies and a product pipeline aimed at treating cancer and infectious disease by helping the body's immune system target diseased cells with precision. The company is developing a portfolio of checkpoint modulators, heat shock protein vaccines, and adjuvants. Its checkpoint modulator programs target GITR, OX40, CTLA4, LAG3, TIM3, and PD1. And its proprietary discovery engine, RetroCyte Display, generates fully human therapeutic antibody drug candidates. We are very grateful for your very generous support of today's webinar, Antigens. My name is Brian Brewer, and I'm Director of Marketing and Communications at the Cancer Research Institute. Cancer Research Institute is the only nonprofit organization dedicated exclusively to harnessing the immune system's power to conquer cancer. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, we'll hear firsthand from an immunotherapy expert how cancer treatment is undergoing a revolution thanks to these breakthroughs. After a 25 to 30-minute presentation, we will open the discussion to questions submitted by you. You can pose your question at any time throughout the webinar by typing in the Q&A box on your screen. We will be live tweeting today's webinar and looking for your comments online. You can follow us at Cancer Research using the hashtag CRIWebinar. Today's webinar is being recorded and will be made available for viewing on the Cancer Research Institute website and on our YouTube channel. Now it's my pleasure to welcome today's expert speaker. Dr. David Reardon currently serves as Clinical Director of the Center for Neuro-Oncology at the Dana-Farber Cancer Institute and previously served as the Associate Deputy Director of the Preston Robert Tisch Brain Tumor Center at Duke University Medical Center. Dr. Reardon is an active researcher with special interests in the design and implementation of clinical trials for neuro-oncology. His work includes using innovative clinical therapies to improve cure rates in patients with brain and spinal tumors, with particular focus on immunotherapies. Dr. Reardon, welcome.
Thank you, Brian. Good afternoon. Hello, everybody. I'd like to, first of all, start off by thanking the Cancer Research Institute, as well as the corporate sponsor, Antigens, for their support of this webinar. There's a lot of material to cover here. I'm going to move pretty quickly, but I look forward to your comments and questions at the end of the presentation. It's a very exciting time in our field, and I think there's a lot of activity here for brain cancer patients to be very hopeful and encouraged by. So just to start off, I have some affiliations where I've served on advisory board or speaker bureau for companies, and my laboratory gets some research funding from a few companies listed here. So what we're going to spend the next 25 minutes or so running through very quickly background information on the primary type of brain cancer that affects adult patients called glioblastoma, and then jump right in and talk about the exciting work that's evolving quickly regarding immunotherapy treatments for brain cancer patients. And I'm going to focus on most of the where most of the research is being applied, and that's for the development of cancer vaccines as well as the use of checkpoint inhibitors. At the end, I'll mention very briefly a comment about combination therapies, because I think this is where the future to optimize these treatments and really have a significant impact will come by putting these treatments together into rationally designed effective combination therapies. So glioblastoma is the most common type of cancer in the brain, but this listing on this not readable slide highlights all the different types of cancers that can arise in the brain. There are 6 major types, most recently classified by the World Health Organization. These are highlighted in blue. And here where the arrow indicates is glioblastoma. So one of many, but certainly stands out in terms of its overall frequency. This pie graph shows the different types of cancers that arise in the brain, and I highlight this slide because it makes the point. Most of the cancers that arise in the brain arise from the supporting structure of the brain. If you think of it like the framework of a house that's being built, this supporting structure is called the glia. This is not consisting of the nerve cells that do our thinking and remembering and interacting with our body and our environment. The glia is simply the supporting architecture of the brain, but all of the major types of brain cancer highlighted here are glial tumors that arise from the supporting architecture. And you can see from the red part of the pie that glioblastoma is the most common subtype, accounting for over half of these tumors, actually. This is a typical MRI scan of a patient recently diagnosed with glioblastoma in our hospital. This MRI scan on the far left are pictures taken from the bottom of the head, moving slowly up, slice by slice, that move from the front of the head to the back and the left to the right. And here we're about up in the middle of the forehead level, and all of the normal brain appears as this gray material. The normal spinal fluid space in the middle of the brain is black, but you can see in the back corner here on the left side of the brain is this white area of abnormality. That's contrast uptake that's highlighting the cancer on this MRI scan. The other two pictures here are taken from as if the patient was looking straight out of the computer monitor at us, and you can see the back left corner of the brain having the same abnormality, and then a side-to-side picture, again showing us in 3 different planes how the tumor appears. And this type of imaging is very helpful for our surgical colleagues to map out and to resect the tumor. Highlight the frequency of brain glioblastoma, this specific type of cancer. There are about 13,000 cases in the United States each year that are diagnosed. Similar number in Europe and in Asia. And this graphic highlights that these cancers, although they can occur at any age, and I've treated patients in infancy and young childhood all the way through adulthood, but the most common age, because they increase as we get older, if we follow this blue curve, this is glioblastoma, they increase in frequency, and the most common age in the 6th to 7th decade of life. The top curve is for all brain cancers, and we see that they actually all increase as we get older during life. This slide just highlights the major categories of brain cancer that we've traditionally thought of them based on how aggressive the cells appear under the microscope. When a tumor has been removed surgically, it's studied very carefully in the laboratory, and based on how aggressive the cells appear, the pathologist will grade the brain cancer on a scale of 1 to 4, with 1 being the least aggressive appearing cells and 4 being the most aggressive. And unfortunately, what we see in terms of the proportion of all brain cancers, with increasing grade or increasing aggressiveness, we see the frequency of these go up, with the most common subtype, grade 4, glioblastoma, accounting for over half of all of these cancers. This slide just highlights how these cancers appear under the microscope to show the difference in the features that we look for to categorize very carefully the type of cancer that we're dealing with and how we've traditionally classified these. We also have learned a lot over the last several years about the genetic mutations and abnormalities that characterize these tumors. And over the next few years, we predict that using these mutations is actually going to be the way that we move to classify these tumors going forward. So we'll be thinking less and less about how they might appear microscopically and more about the genetic abnormalities that define and predict how they're going to behave. A question I get very frequently in the clinic is, why does brain cancer occur? Why did this happen to me? And unfortunately, I don't have, and nobody has, a very good answer to that question. There's been a lot of research that's been done and a lot that's ongoing to look at either inherited or familial factors as well as environmental factors that could contribute to the development of these cancers. Fortunately, we know only a very small percentage of these cancers occur or run in families. The vast majority have no genetic or inherited predisposition, and I always reassure my patients that just because this cancer was diagnosed in you, the likelihood of this happening in your immediate family members, your brothers and sisters, your children, is essentially that of the general population. There really is not a significant risk to them. Similarly, looking at environmental factors that can contribute, very good studies have been done and continue to be done, and we haven't come up with good links or causative factors that people are exposed to, with one exception, and that is when a radiation has been previously applied to the head or scalp, there is an increased risk of brain cancer. But fortunately, everything in the environment does not seem to be linked, including cell phones at this point. So I use my cell phone quite a bit and reassure my patients it's not going to cause them brain cancer. Let's switch gears and talk about current state-of-the-art treatment for glioblastoma. The state-of-the-art is summarized on this slide, and it begins once the patient is diagnosed with a maximum safe surgical resection of the tumor. The patient heals up after surgery is done for about 4 weeks and then begins the radiation phase of treatment, highlighted in the red arrows here, where radiation is given Monday through Friday for 6 weeks. And during that time, a daily small dose of chemotherapy called temozolomide, or TMZ, the patient takes that every day during the 6 weeks. Then they have a 4-week break to recover from this initial phase of intensive treatment and move on and take monthly cycles of chemotherapy, the same chemotherapy, temozolomide or TMZ, but during this phase of the treatment, which goes on for an additional year, the chemotherapy is taken for 5 days at the beginning of each month. Now, this standard of care for treatment was defined 10 years ago in 2005 by a study that compared patients who received this treatment, including chemotherapy with radiation, to what had been the standard of care at that time, radiation therapy alone. And the study showed that patients who had the addition of the chemotherapy had an improved outcome. The time it took for the tumor to recur, or progression-free survival, was increased by just about 2 months, and the median overall survival, or OS, of the patients increased by about 2.5 months. This was far from a home run, but it was certainly an improvement over what had been the standard treatment with radiation therapy alone. But unfortunately, most patients, the tumor will recur after the standard of treatment. And as I mentioned, it recurs typically, the average time is about 7 months after diagnosis, which puts things right at this red arrow here during the course of treatment. So to highlight how critical a need we have for better treatments, the average patient with our current standard of care is already developing recurrence or relapse of their tumor about halfway through their planned course of treatment. Now, given that most patients will recur, we've worked very hard over the years to come up with better treatments to use at the time of recurrence. And this bottom half of the slide shows a number of different trials that have been analyzed in meta-analyses through different cancer cooperative groups in the United States, Europe, and Canada. And unfortunately, the percentage of patients with these treatments at the time of recurrence who are able to maintain control of the treatment for control of the tumor for 6 months or longer, this 6-month progression-free survival percentage, is unfortunately only about 10%. So we desperately also need treatments that can help patients once the tumor has recurred and is growing after standard therapy. This is what we have available to help our patients in 2015. These are the approved agents in the United States for glioblastoma treatment. We have a couple of different variations of chemotherapy, the nitrosoureas, BCNU and CCNU, and a variation of this that can be implanted into the surgical resection cavity called Gliadel. We have temozolomide, the chemotherapy that I described being part of the important standard of care now. And then we have the prototype of the cancer-fighting agents that block the blood supply of the cancer, the anti-angiogenic agents, bevacizumab, otherwise known as Avastin. These 4 agents are what we have that's approved for brain cancer patients in 2015. Now let's talk about the immune system, because I want to switch and begin to think and talk about how we can use the immune system, the body's natural defense system, to fight cancer. And this slide is a simplified version of how the immune system, the body's normal defenses, normally work in the body. There are 2 main arms of the immune system, one called the innate arm and one called the acquired arm of the immune system. And they both work together in concert to help protect us from anything foreign that could be invading or attacking our body, like an infection, for example. The innate immune system has been around for a very long time. This is the evolutionarily old version of the immune system. It consists of 2 components, physical barriers, as well as components within the bloodstream that are mobilized very quickly, immediately, in a non-specific manner to attack what's perceived as foreign, potentially harmful, and try to prevent that from invading the body and causing damage. The protection that this provides, this part of the immune system, is typically not very long lasting. This has been around in part of most species, plants, insects, and invertebrates. As species have evolved and become more advanced, the immune system has also advanced, and the acquired component of the immune system is part of most vertebrate species or more evolutionarily advanced species. And this also has 2 arms. B cells, which generate antibodies to attack very specifically a foreign material, as well as T cells, specific cells of the immune system that in and of themselves can attack a potentially harmful or foreign invading material. The acquired immune system is adaptive, meaning that it responds and generates a response against a specific organism or foreign material. It's very specific. It only attacks that particular thing that it recognizes as abnormal or foreign. And it has the capability of providing a long-lasting protective component, because the immune system Mother Nature designed us with has memory. Once the immune system, the acquired immune system, is stimulated, it never forgets. That part of the immune system stays with us forever. And if that infection or foreign material were ever to come back to the body, the immune system against that specific material would activate and attack it before it had a chance to harm us. That's how things normally work in the body for the immune system. Now, trying to use the immune system to fight cancer has been a dream for quite some time, going back 115 or more years, back to William Coley, who was the first clinician scientist to try to use the immune system to fight cancer. This was back in 1893. Since then, for many years and decades, a lot of work has been done, and until very recently, not a lot of positive results were achieved. Things were fairly disappointing with try to learn about the immune system and how to use it to fight cancer. Until about 2010, in the last 5 years, we've learned a lot and capitalized on that research and learning in order to help us develop effective weapons and therapies to fight cancer. And this next slide highlights some of the important advances. In 2013, immunotherapy for cancer was defined as the breakthrough of the year. The American Association of Cancer Research, the leading cancer research society in the world, developed its own journal dedicated to immunotherapy for cancer called Cancer Immunology Research. And in the last 5 years, we've had 5 different immunotherapies approved by the FDA, as well as abroad in other countries, for a variety of different cancer indications. And more approval with these medicines are likely forthcoming in the next few years, we predict. So let's switch and talk about immunotherapy for brain cancer specifically. And there are a couple of challenges and concerns that are, I think, unique or certainly worthy of consideration relative to other types of cancers. The first of these is the location of brain cancer being unique and within the brain itself. When I was in medical school, I was taught that the central nervous system, or the brain, is an immunoprivileged area that is protected from the immune system and does not interact with the immune system. So for many years, immunotherapy for brain cancer was felt to be a mission impossible, that because of this immunoprivilege, we wouldn't be able to use these therapies for our patients. And this data goes back for many years, back into the 1940s actually. But now with much research that's been done, we've learned that at least what I was taught in medical school is not quite accurate. And the immune system does indeed interact very effectively in the body with the immune system in the brain, that there's a very dynamic and effective interaction that allows the immune system to work very effectively within the brain and central nervous system. Another very important consideration with brain cancer is that this is one of the most complex and heterogeneous of all cancers. What I spent the better part of 20 years studying and thinking was a single entity, glioblastoma. We now know that there are 4 major subtypes of glioblastoma based on looking at the genes that are expressed by these cancers. They can be classified by this gene expression profiling capability into 4 major subtypes. And then we also have learned from some of the elegant, more recent research that's been done that brain cancer can be heterogeneous, meaning within a given tumor, there can be a lot of variability within the cancer cells, and that variability can change and evolve over time as well. To highlight the heterogeneity within a given cancer, this busy slide shows elegant work that was recently published looking at single cells that were isolated from 5 different tumors resected out of the operating room, and individual cells were separated out and analyzed. And with that single cell analysis, that highly technical analysis, we're able to see very significant differences in many different aspects of the cells of these specific tumors, including the fact that within a given tumor, there are many different subtypes within that given cancer, as highlighted on this slide. So that heterogeneity within a cancer has been a very significant concern when we think about standard traditional chemotherapy, radiation, and biologically based targeted therapies. The more heterogeneous a cancer is, the less likely it's going to be able to respond to treatment. So here we see the higher the heterogeneity, going from 0 to 100, the likelihood of durable treatment success goes down in a proportional way with traditional therapies. But if we stop and think about the possibility with immunotherapy and the way the immune system is designed to attack anything foreign in a very specific way, the exact opposite trend can occur actually, where immunotherapies, the more complex or heterogeneous a cancer can be, the higher the likelihood the immunotherapy may be able to be effective. It can also be effective against not very heterogeneous or very simple cancers, that's true as well, but in fact, one could hypothesize that the more complex the cancer is, the more likely the immune system could be successful in attacking it. Another very important aspect of cancer cells is that they have adapted mechanisms to protect themselves against the immune system. A general principle of cancer cell fitness, if you will, is that cancers have evolved to dysregulate normal physiologic protective processes that we all have in the body to make themselves stronger and to achieve a survival advantages. And there's several examples of this that I've listed here. Cancer cells can upregulate normally present DNA repair enzymes to protect themselves. They can overactivate pathways within cells that normally regulate and control growth and survival. They certainly can upregulate the ability to make blood supply and new blood vessels to feed themselves. And the last and important example relative to immunotherapy is the ability of cancer cells to express a number of different molecules that suppress the immune system, turn the immune system off as a strategy to protect themselves so the immune system does not attack them. And this immunosuppression of cancers is certainly present in brain cancer, and I think of it like the different layers of an onion, with the cancer very much in the middle here, but these multiple layers of protection all around the perimeter of the tumor to wall it off and protect it. There are certain molecules that affect the bloodstream to weaken the immune system, and then a whole host of different molecules that are present within the microenvironment of the cancer itself that function to protect the tumor and turn the immune system off. So a way to think about this in reality of how things work in the normal system and then relative to cancer, if we think about what happens when our body is exposed to a foreign material like an invading organism, what typically happens is that the immune system is activated, and initially the immune system is turned on in a very proactive and activated way. But before very long, the immune system counterbalances with time, so that normally the activity of the immune system is downregulated so that we get a nice effective response to protect us, but not something that's going to be overactive and cause damage to normal cells in the body. So these balancing suppressive molecules normally occur in our body to help downregulate the immune system and protect us from overactivity and damage, otherwise known as autoimmunity. And this is the part of the immune system that cancers have over-evolved and developed to exploit and protect themselves as a strategy to turn off or evade the immune system. So here, in this example, where the foreign target is a cancer molecule, the immune system, instead of having this robust activity that we normally see, the activity is diminished, and a whole host of suppressive molecules and factors are secreted by the tumor to protect itself and keep the immune system in a less active state. And what we've learned from much of the research that's gone on over the last few years is that the key molecules that normally work to protect us from the immune system getting overreactive are called immune checkpoint molecules. And I think of these as a simplistic analogy of kind of the brakes and a clutch in a manual transmission of a car. These molecules normally turn down the immune system to keep it from causing damage. Cancers increase these particular molecules, the two of the ones that have been most studied, called CTLA-4 and PD-1, and its binding partner, PD-L1. Many cancers upregulate these molecules as an important strategy to protect themselves. And this is a table showing the percentage of different types of cancers that have upregulated the PD-1 or PD-L1 molecule as a protective immunosuppressive molecule, and glioblastoma clearly being on the list here. And a more recent study just published earlier this year showing all of this brown material is the PD-1, PD-L1 expression by glioblastoma tumors as we look at the cells under the microscope, showing that over 90% of the tumors have a very high rate of this immunosuppressive protective molecule. So there are important considerations for immunotherapy as we think of and apply it for brain cancer, but there's a lot of excitement and a lot of enthusiasm in moving forward with different treatments. And I'm going to highlight now some of the work ongoing in the clinic with vaccines, as well as using inhibitors of these immune checkpoint molecules to treat and help brain cancer. So let's start with vaccines. How do they work? Well, remember, as we grew up, we were exposed to a variety of different vaccinations to protect us from different types of infection and help strengthen our immune system. Should we be exposed to these infections, our immune system would be primed and able to attack it. And we can see with these vaccinations that have evolved over the years, the percentage of improvement and reduction in death rate associated with the infections associated with this prophylactic vaccination strategy. Cancer vaccines work in a very similar manner where we expose the body and the immune system to components of the cancer, whether it be peptides or proteins or DNA or even a lysate or a mixture of the cancer cells themselves. Inject those and use those to sensitize the immune system in the body to develop a response specific against these molecules associated with the cancer. There are a lot of different targets for brain cancer that we can use for vaccines called tumor-associated antigens or peptides. And the other strategy I mentioned of just taking the tumor out and using that, making a lysate of that and using that to sensitize the immune system. This work has been ongoing for over 10 years now in brain cancer and showing some promising, encouraging results. But what I want to focus on now are two studies that have recently been presented at major cancer meetings that show some encouraging results for the first time of vaccine therapies for glioblastoma. These were two randomized placebo-controlled trials, meaning that they were very stringently and carefully conducted studies to see if these vaccines, one called ICT-107 and the other called rindapepimut, could have a benefit and help brain cancer patients. The first study, the ICT-107 study, was conducted in newly diagnosed glioblastoma patients. This vaccine consists of 6 different peptides that are upregulated and expressed on glioblastoma tumors. And this cocktail of these peptides is used to sensitize special white blood cells from patients called dendritic cells, or DCs. And once these dendritic cells are sensitized to the peptides from the cancer, they're given back to the patient as an infusion. This study was a randomized placebo-controlled study for newly diagnosed GBM patients in a 2-to-1 manner, meaning that for every 3 patients enrolled, 2 got the vaccine, along with standard radiation and chemotherapy, and 1 patient randomized to get a placebo, along with standard radiation and chemotherapy. And the key results of this study showed the vaccine was very safe, and that's true of basically all of the cancer vaccines that have been evaluated to date, and that we saw some modest improvement in the time it took for the tumor to reactivate or grow, called the progression-free survival, but unfortunately no significant survival benefit. However, when the data from the study was broken down and we looked at the patients who were predicted to most likely have a benefit from the vaccine, that subset of patients actually did have a significant improvement of both the progression-free as well as the overall survival with the vaccine. Studies also done with this trial showed the vaccine preserved quality of life for our patients, and that the vaccine generated important immunogenic responses within the immune system. So this is the slide showing in the blue line the time it took for the tumor to activate, or progression-free survival, among the vaccine recipients. The further it is to the right is the higher percentage of patients at a given time point for the tumor to remain under control. So we see the blue line being shifted over to the right means a higher percentage of patients over time remained under tumor control than the control patients. Unfortunately, the survival curves really didn't show a significant separation between the treatment arms when we looked at all of the patients enrolled on the study. However, when we broke down and looked at the predicted patients who were most likely to benefit, here we saw what looks to be a pretty robust level of activity with this vaccine. A 15.5-month improvement in time to progression with the vaccine compared to the control. That's a very significant improvement. And for survival, the blue line here clearly showing an improvement in survival for the vaccine patients. So this was the first study to show some encouraging results in a randomized placebo-controlled manner for vaccine, and the vaccine showed about a 2-fold increase the targets the vaccine was intended to attack. The second study that showed us a survival improvement was targeting a mutant receptor on tumor cells called EGFRV3, and the schema for how this appears on the cell of cancer cells is depicted in this slide. But just to summarize, this mutant molecule is present in 30% of glioblastoma tumors. And because it's never found on normal cells, it's only present in the cancer cells, it's a perfect type of target to think about for using for immunotherapy. And when I was at Duke University, worked with many excellent colleagues there, including John Sampson, who developed a protein vaccine against EGFRV3, and we tested it in early clinical trials for patients and showed in the blue lines here, the patients who got the vaccine had a significant benefit compared to historic patients who did not get the vaccine. So these were early studies that we were very encouraged by. We were also encouraged by the fact that when we looked at tumor material that was removed after the patients had progressed on the vaccine therapy, although they all had the target EGFRV3 readily detectable on the tumor cells prior to vaccination, nearly all of the patients lost the V3 mutant peptide when the tumor came back. And this was, I think, proof of principle that the immune system was able to eradicate at least that component of the tumor that had the EGFRv3 positive cells. Since this data was generated, this vaccine has gone into a large clinical trial called the ACT-4 study. This is actually completed accrual. This is for newly diagnosed patients, where half of the patients were randomized to get standard therapy plus the EGFRv3 vaccine called rindapepimut, or now known as Rintega, and half of the patients got standard of care plus placebo. We have results, preliminary results of this study, hopefully by the end of the year. But in the meantime, we designed a study to test this vaccine in patients with recurrent glioblastoma. And here we, at least at that time, worried that these patients, because they were recurrent, may not respond very well to a vaccine because their immune system was likely fairly weakened at that time. But nonetheless, because the vaccine was showing some positive results in the newly diagnosed patients, we thought we should test it and see how it worked in patients with recurrent tumors. So we designed this study called the REACT study. And this study I presented along on behalf of my co-investigators and colleagues for the study at the American Society of Clinical Oncology meeting just a few weeks ago in Chicago. And the data for this study looked very encouraging and exciting. The trial we designed here, we randomized patients after they had recurred with their tumor to get Avastin plus rindapepimut, the vaccine, or Avastin with a control placebo vaccine. The study, the key results are summarized here. Again, the safety was quite strong. There were no significant side effects associated with the rindapepimut vaccine. And here, for the first time, we saw patients who got the vaccine had both a progression-free as well as a survival benefit. And this was a historical achievement. This is the first time in the history of treating glioblastoma that a placebo-controlled randomized study has shown the study population benefits from the use of an immunotherapy, in this case, rindapepimut, the EGFRv3 vaccine. So we have our first very strong piece of data to confirm that immunotherapy can help brain cancer patients. On this study, we saw a higher percentage of patients whose tumors improved on MRI scan, the radiographic response rate, 30% versus 18%. But the vaccine patients highlighted in green, what was very encouraging is not only did the tumor get better, but it stayed better. They had a much more durable benefit associated with the response. Similarly, a higher percentage of patients on the vaccine were able to come off corticosteroids, which is a major positive step. Steroids have a lot of side effects and complications for patients, and they all hate being on them. The vaccine also improved progression-free survival, or the time it took for the tumor to start to grow again, but in a fairly modest way, not a dramatic way, but nonetheless an encouraging way. But what was most encouraging, what was most exciting, was the fact that patients who got the vaccine lived longer. And this was the first time we've seen an immunotherapy able to improve survival for patients. It's not a home run. It was a modest improvement of just basically 2.5 months of living longer. But it's a proof of principle, an exciting first step that I think we can certainly think about in a very positive way, building and going forward on. The vaccine was associated with a high rate of reactivity and generating of anti-tumor immune responses, even though the patients were recurrent and had received a fair amount of prior treatment. So in terms of wrapping up with vaccines, it's a very exciting time, and many of us are thinking about how we can build on these early results and try to make them more effective. We've started a treatment here where we are characterizing the mutations associated with each individual patient's tumor, called Neoantigens, and generating a tumor-specific vaccine for that individual. This is work I'm doing here at the Dana-Farber with my primary collaborator, Kathy Wu, who's developed this technology. And this takes advantage of the mutation frequency we know that happen in cancers. All different types of cancers listed on this graph here, and the number of mutations associated with each cancer highlighted on the y-axis here. We see glioblastoma has a pretty high rate of mutation associated with any given individual tumor. And Kathy and our group reasoned we can take advantage of that therapeutically by taking the tumor out, analyzing and characterizing these mutations, and generating a personalized individual vaccine to give back to the patients as their treatment. And this trial is a very early trial. It's a proof of principle trial where we're looking at the feasibility of doing this. It's never been done before for brain cancer. We do have a trial ongoing at the Dana-Farber for melanoma with this approach, but we're applying it for brain cancer. And this is the design here where patients have surgery. We collect the tumor, we begin to generate the vaccine while they're getting radiation therapy and recuperating. And then they get a series of injections, 5 over 1 month, and then 2 booster injections over the next few months as part of their treatment. We've treated three patients, about to treat a fourth, and a number of others coming along here. So it's too early to say, but we're hopeful this will also show some benefit for our patients and another step to build on. Let's wrap up and come back to the important aspect, though, of immunosuppression and how tumors evade the immune system by making molecules to turn the immune system off. Remember the graphic I showed earlier where a cancer cell makes these molecules that suppress the immune system and keep the immune system less active. With this research and understanding this, we now know what many of these molecules are that tumors suppress the immune system. There are these checkpoint molecules, and the research that's identified these has allowed to develop therapeutics to inhibit these checkpoint molecules, 3 of which I've listed here on the bottom of the graph, that basically block these inhibitors and allow the immune system to be much more active and actually come back more toward a normal balance state where the suppressive factors are decreased and allowing the tumor, the immune system to be much more active against the tumor. We wondered how well these molecules would work in brain cancer patients because they seem to be working in other cancer patients, and we didn't know how they'll work in the clinic, but decided to test this in the laboratory. And I work with a couple of very talented colleagues here at the Dana-Farber, and we have a mouse model of brain cancer where we've used some of these inhibitors developed by my colleague Gordon Freeman to test in this brain cancer model. And let me just summarize a lot of work that we've done over the last couple of years to look at this important question and show that with at least one of these inhibitors, we're able to long-term cure 50% of the animals. And when we put a couple of these molecules together in a combination, we can push the cure rate up to about 50 to 70%. And we're running low quickly. We're running down on time, so I want to just move through this very quickly. When we look at the animals, the control animals have big tumors and they all die within about a month. With the immunotherapy we use, the tumor regresses and the animal becomes a cured long-term survivor. And when we re-challenge the animals, we re-inject them with a tumor load to see if the tumor will be able to grow. We see that for the vast majority of the animals, the tumor is not able to grow, and that tells us that the immune system has been primed. Once it's primed and memory is established, it can theoretically prevent the tumor from relapsing and recurring, and that's what we've shown in this particular animal model. We've also shown that the treatment causes a very important activation of the immune system by looking at the different types of immune cells in the animals. Now, moving into the clinic, these results and others that other researchers have generated have helped us move these treatments into the clinic to evaluate for patients. And this is the listing of the currently active clinical trials that are using immune checkpoint inhibitors for glioblastoma. There are 5 of them that are currently active in enrolling patients. The targets, the type of study, the patient population that's being studied, the number of patients that are being enrolled, and the design of the study is summarized on this busy slide here. It's too early to generate any results and know how well they're working, but we will have some answers, I think preliminary answers at least within the next 6 months, certainly the next year on these important results. So to wrap up, can brain cancer be treated with immunotherapy? Despite what I was taught in medical school about the immunoprivilege of the brain and the central nervous system, we think this is not so much of an important consideration any longer, and that should not prevent us from using immunotherapy for brain cancer. There are a number of different strategies, vaccination strategies. I haven't talked about cell-based therapies. These are just beginning to be studied and developed for brain cancer, but more will likely evolve with those over the next few years. Overcoming the immunosuppression the cancers generate is a critical step that we need to be better understanding and better developing therapies. And I think our first treatments to do this with the checkpoint inhibitors certainly showing encouraging results in other cancers that we hope we'll see in our patients. And then just to finish up, my personal bias and feeling is that for these therapies to work in a durable way for a high percentage of patients, we're going to have to put them together into combinations where we, in the first step, sensitize the immune system against the tumor and then combine that with a second treatment that can block some of the suppressive capabilities the tumors have adapted in order to allow that immune system response to eradicate the cancer. So that I'll wrap up here, and thank you all for tuning in, and again thank Cancer Research Institute and Agenus for supporting this important webinar, and I look forward to your questions and comments.
A wonderful presentation. I thought very easy to understand. You have a great way of describing what's a very complex science and a lot of advances that are happening very rapidly in the treatment of cancer with immunotherapy. So again, thank you for that. We do have a number of questions from audience members. So the first one was, you showed us that glioblastoma comprises the majority, more than half of CNS or brain cancer tumors, and all the trials that you showed us dealt explicitly with that type of cancer. But there, of course, as you showed us in your first slide, many different types of CNS cancers. Are there immunotherapy trials ongoing for those?
Yes, actually, there are. There a lot of systemic cancers metastasize to the brain, unfortunately, and that historically has been a very challenging development to treat for patients. So metastatic cancers, there are these immune checkpoint inhibitors that I mentioned have shown some exciting results for metastatic cancers. Other primary cancers or cancers that arise in the brain rather than spreading to the brain, there are clinical trials ongoing for those other types of cancers as well. The glioblastoma is the most common type and the type with the greatest unmet need at the moment, and that's where most of the research effort has focused. But certainly other types of cancers are also of the brain are also getting we're learning more about how to use immunotherapies for those cancers as well, and there's a couple of clinical trials that have already started with more to come.
That's great, and I'm glad that you touched on other types of cancer that metastasize to the brain. We had a number of questions there too, so it's good to hear that there's some work being done in that area as well. Something that you didn't touch on was how these are administered. So you spoke of kind of the immunoprivileged myth, that fallacy, and how that's since been shown not to be the case, that the immune system does have activity in the central nervous system. But another thing that we hear a lot about is the blood-brain barrier, which is, I think, related in some ways. So the question is, how are these immunotherapies administered intravenously, intracranially? Are they injected into the brain? How does that happen?
So our immunotherapies so far for our patients are administered, vaccines are typically administered as a simple injection right under the skin, usually near lymph nodes, which are the major structural organs of the immune system, where a lot of the immune system cells are localized and organized and develop their functional activation. So it's a simple under the skin injection for a vaccine near these lymph nodes, typically either under the arm or in the groin area. That's a very small needle and very easy and well tolerated by patients. The other immune checkpoint inhibitors and cellular therapies that are being developed are typically administered intravenously directly into the bloodstream. And by that route, they're very able to effectively penetrate through the blood-brain barrier and get into the brain and the tumor to help fight the cancer.
All right, thank you. So with so many, I thought the Rindopetamute trial that showed for the first time that immunotherapy is effective in brain cancer was really good to see. And with the plethora of trials now that are testing the checkpoint inhibitors alone and in combination with other therapies, there seem to be a lot of options now for patients, at least in the clinical trial phase. And so the question is, With so many trials available, and you mentioned that the future is going to involve looking at the specific neoantigens, the unique mutations in each patient. Should patients have their tumors, when they're surgically removed, saved and pathology done on them to test them for these markers? Is that something they need to ask, or is that something that will be done automatically?
Well, more and more, particularly at academic centers, when tumors are collected in the operating room, they are processed so that we can test for them at least the most common types of mutations. And those are the ones that many of the therapies are particularly, at least at this stage, beginning to focus and target on. So yes, that's typically being done on a routine manner. And most of the academic centers also are very careful to use as little possible of the tumor to make the diagnosis or what's removed out of the operating room, and then to be able to preserve and to save as much as possible of that tumor in a liquid nitrogen or frozen tumor bank, and keep it available for future research and study that could potentially help patients. We also know that though, because the tumor can evolve over time, that if the tumor does come back, there may be differences within that tumor at the time it came back from when it originated. So if we're talking about an immunotherapy, probably getting a tumor sample as close to the time of the immunotherapy will be most helpful.
Okay, thanks very much. On some of the progression or the overall survival charts that you showed us, we saw in a small percentage, it looked like less than a quarter of patients, you see this long tail kind of moving along. And this is something that we've seen in the trials with ipilimumab and nivolumab and pembrolizumab, where patients who respond to the immunotherapy continue to respond for a duration of time. Do you anticipate that those patients who are responding will continue to respond, and do they require follow-up treatment once the trial is ended? Will they need to continue to receive these therapies, or does that response just happen naturally in the body?
Well, that's a great question. And one of the most exciting things about immunotherapy is that it provides a twofold capability that we've never really had before for cancer treatment. First of all, the immune therapy activates pretty quickly and can attack an existing tumor and get rid of that. And if the immune system is primed properly, any cells that happen to survive, be able to lay dormant and then potentially reactivate that could otherwise cause a recurrence. Once they started to do that, the immune system would reactivate and attack those cells to prevent a recurrence. So the immune system can both kill off an existing tumor as well as prevent a relapse in the future. And that's where that tail of the curve, where we see a percentage of patients who are long-term survivors that's never been achieved before for something like metastatic melanoma, is so very exciting. We now have patients 5 to 10 years out with what had previously been a uniformly fatal cancer, who are now surviving 5 to 10 years with no sign of the tumor coming back, effectively cured. Our challenge is to develop these therapies further, optimize them, begin to bring them into combinations where we can raise the percentage of patients from a relatively small percentage to a much higher percentage of patients to get into that long-term, effectively cure situation.
Yeah, there's certainly a great deal of work that remains to be done, despite the excitement of the successes we've seen in the clinic with especially the checkpoint inhibitors. I know our organization and all the scientific advisors that work with us in our global network of researchers who are focused on immunotherapy all agree that there's still a great deal of work that needs to be done, and we're very grateful that there are people like you who are out there doing it. So another question we have before we wrap up is, you showed a few trials where bevacizumab was kind of used in combination with, for instance, checkpoint inhibitors. And I guess it's two questions. One is that because this is the standard of care and that this is what patients should be receiving anyway? Or is there a rationale for combining the two? Do the two produce a synergy where they enhance the effect to the patient when used together?
And the answer to that question, to both of those questions, is yes. The use of bevacizumab, the prototype of this cancer therapeutic to block the ability of the cancer to make a blood supply to feed itself, try to starve the tumor. That is the standard of care for recurrent glioblastoma in the United States. It's a standard of care that does help patients in a meaningful way, but unfortunately is not durable. So that is what we offer to patients. But very excitingly, we know from some of the ongoing research that the molecules that Avastin is designed to target are very critical contributors to what the immune system uses to protect itself, what the cancer uses to protect itself from the immune system. It's part of this immunosuppressive cloaking device or ability of shielding itself that the cancers exploit and utilize. So there is a scientific rationale as well that by blocking angiogenesis of the cancer, we may also be able to improve the effectiveness of immunotherapies. And we have trials ongoing now for brain cancer. There's already been one published for metastatic melanoma by my colleague Steve Hody here that does show that hypothesis may prove to be correct and helpful. So we're very excited about that option and combination approach as well.
It's fantastic. A question, when we were talking just a moment ago about whether or not it's standard operating procedure to have tumor tissue samples preserved at the time of surgery, you mentioned specifically at academic centers, but many patients are treated at community oncology centers. Are practices different there? Should a patient who has brain cancer perhaps consider looking for treatment at an academic center rather than a community center?
It's always a bit of a tricky situation, but I think in general, centers that have a dedicated multidisciplinary team, and treating brain cancer is very much a team activity here. Neurosurgeons, radiation therapists, oncologists, neuroimaging people, the pathologists, very important roles that when you have that team in place, patients can do better, and there are more options that may be available to them because those are the centers that often are participating in the research. But patients, unfortunately, with this disease, sometimes because of the way things that happen and evolve, they get sick very quickly. They need to be taken care of acutely in the community. But I have a lot of patients, once that acute phase is done, who then come and see us or come to an academic center. And we can work to collect the tumor that was operated on locally, bring that in and do at least an analysis of the genetic abnormalities. That's certainly capable, even if the tumor wasn't resected here, that we can use the material resected locally to provide that analysis.
All right, great. Well, unfortunately, that's all the time for questions that we have. And so we'll start wrapping this up once again by thanking our generous sponsor, Agenus, for supporting today's webinar. We couldn't do it without you, so thank you very much, Agenus. Also, to let you know that our next webinar and our final webinar in the Cancer Immunotherapy Month series is Getting to Cures the Next 5 Years in Immunotherapy. So we've heard a lot about all the progress that's been made to date, and many of our presenters so far have indicated some of the future directions, but our next webinar with Dr. Jed Walchuk will really neatly summarize where we can expect to see the field move within the next 5 years. So that's definitely something that patients and caregivers, I think, would want to see. You can register for that at going to cancerresearch.org/webinars. Also, some of you had asked during today's presentation how you could enroll in a clinical trial or gain access to some of the treatments that Dr. Reardon described. I would suggest that you could go to www.theanswertocancer.org, which is a website that the Cancer Research Institute has built specifically for patients and caregivers who are looking for information about immunotherapy. And the site features a phone number where you can contact a navigator who will help you locate a clinical trial for which you or someone that you're caring for may be eligible. So I strongly encourage you to go to theanswertocancer.org about that. And with that, I'd like to thank you again, Dr. Reardon, for a very excellent webinar, and I'd like to thank all the participants who watched today and those who will be watching via our YouTube and CRI website channels. In just a moment, we'll be closing the room and you'll receive a survey. Your feedback is especially helpful so that we can improve these webinars and provide the best service to you today. So thank you again all very much. And if you'd like to learn more about the Cancer Research Institute, come to our website at cancerresearch.org. Thank you.